All right, everybody. So welcome to intro video part two. So today I'm going to be talking about pipetting and scientific measurement. Um, so, well, before I get started on that, just want to run through this one more time. So again, this is the central dogma of biology, or as I like to call it, the flow of life. Information is stored in DNA. That DNA is transcribed or copied into RNA as an intermediate, and then the RNA is translated into protein. And those are the three levels of life that we'll be studying in this class. And we'll talk more about that as we go along. I just want you to start having that process memorized. So anyway, so what do we do in this class? Essentially what we do is we take stuff out of little tubes like this. So I'll stop my share for a second. All right, see this little, there's a microcentrifuge tube. Here's another microcentrifuge tube. We take stuff out of some microcentrifuge tubes and we put them into other microcentrifuge tubes. And so, yeah, I'm sure that sounds like a lot of fun, but I promise it is. And you can see that these things are pretty small. And to do that, we have to do a certain type of measuring. And so, if you've cooked before, you probably have measuring implements that you've dealt with that kind of look like this, but obviously you can't use something like that to measure volumes like this. So what do? We use these things called pipettes. And so right here is an example of one pipette. Let's go back to the slides for a second. And so we're also going to be using things like graduated cylinders and scales, but the pipettes are the big ones. So as we look at a pipette, and so you'll have to kind of look at both the little screen and the big screen. The pipette is essentially like a specialized plunger. And so there is like a button here at the top. We call that the plunger. That's the main thing that we use to draw stuff up into the pipette and push things out. So the basic process of pipetting is, stop our share for a second. So you have the pipette. There's this little window in the front that shows you your volume. We'll talk more about that in a second. That faces away from you. And you can see there's like a little grip thing that shoots out. Wrap your hand around it like that. You have your thumb on the plunger like that, and you push down like this. And so when you push down on the plunger, you're going to feel some resistance. So let's try to get that in our view a little bit better. We hit this resistance point, and then you can actually push harder, and it'll go down even further. So when we go to measure things, and the first thing we do, all pipettes have tips that you put on them. You never use a pipette without a tip because we don't want any liquids on the inside of this thing. We only want the liquids to be down into the tips. So what we do is we take our pipette, we go open the tube that we're gonna draw stuff out of, we go down, we push our plunger down to that first resistance point. We put our tip down into our liquid. See the tip is down in the liquid there. Then we draw up. Okay, you can see now, you can see that there's liquid down in that pipette tip. So then what we do is we get our other tube that we're going to go into, go down into that tube. We watch what we're doing here. We don't just kind of like do pipette like around somewhere. We're watching where the tip is. We're going to put that down into the bottom of the microcentrifuge tube, and then we're going to push out the liquid. And then you can push further, and that extra step beyond the resistance point just pushes a little bit of air out of the tip so that you're getting all of your liquid down in there. And so if you can see down in there, you can see there's a little bit of liquid down in the bottom of the tube. So to change the volume, right? So how much we pull, how much liquid we pull up into that tip, we rotate knobs. Each pipette has knobs up at the top of it like that. We don't rotate the plunger. We rotate the knob. Okay. And so as you rotate that knob, you can kind of see it there. This number changes. And so lefty loosey brings the volume up, righty tighty brings the volume down. And so you can see as I'm going up here, this number is getting bigger. And so that number corresponds to different volumes. And each type of pipette, check this for a second, each type of pipette can do different sets of volumes. So we have a P20, a P200, and a P1000. And so this one can measure up to 20 microliters. This one can measure up to 200 microliters. And this one can measure up to 1,000 microliters. And we'll talk more about what microliters are in a second. The P20 and the P200 both use these tips like this. The P1000 uses different tips, and I don't have any of those right here with me. So we change the volume with the knob. We do our pipetting with our plunger. Let's go back to our slides. All right, and so, like I said, we're going to be plunging liquid in and out. Whenever you pipette, you want to do that relatively slowly. If you let the plunger flick like this, that's really bad. And what can wind up happening is that that flings liquid up inside of the pipette tip, and we don't want that to happen. So always pipette slowly and thoughtfully. One other thing, I kind of did this one off camera. When we put the tips on to the pipette, we leave the box on the table. We give it a little gentle tap to help make sure it's stuck on there. And then we have this other button that was sort of hidden by my thumb when I had my hand on here like this. So this button is the tip ejector. And so what that does is it ejects the tip off of the pipette. And don't fling it at your computer like I just did. That we have specialized containers that we eject our tips into. And don't put it at someone's eye. You'll put your eye out, kid. So yeah, those are the main parts of the pipette. Again, these are the different types of pipette, the P20, the P200, and the P1000. Like I showed you before, the different pipettes have different volumes written on the top. You can also tell the difference based on looking at the bottom. The P1000 has a real fat bottom. The P200 is sort of intermediate, and the P20 is kind of thin. The numbers, the P20, the P200, and the P1000, those are your upper limits for what you can measure, and never make your dial go above that. If you crank your dial out, it can really screw up the pipette. Conversely, they each have a minimum volume that they reliably measure. And so the P20 can reliably measure about 0.5 microliters to 20 microliters. When we look at the window and see the number on the inside, we have two black numbers and one red number. The black numbers are the whole digits, and the red number is the decimal place. And so we never want to see that one above 200. And typically we don't want to do it below 005. Okay, so those are sort of our limits there. For the P200, all the numbers are black. And so essentially we're looking at the whole number. So whatever we see there. So if it's 173, it's 173 microliters. And in that last position, you can finagle it a little bit to get some decimal places in there, but it's not super reliable at measuring decimal places. So we only, with a P200 or P20, you can get like 2 decimal places pretty reliably. With a P200, you're sort of stuck to 1 decimal place. You can kind of estimate it, but it's not great. Then with the P1000, again, we have 3 decimal places. This one, the first digit is red, and then the next 2 are black. And that one, I don't know why they do that with this, but I find it a little confusing. So basically whatever number you see here, you multiply by 1,000, or sorry, by 10. And that gives you your thousands. And so right now this is saying 3030. So that means we're measuring 300 microliters. The red decimal place is really only useful when you get to 1,000. Okay, so let's bring that up here. Okay, so we're at 1,000, just to show you that here, right? So we're at 100. So that's showing 100 here, but we multiply that by 10, so that's 1,000 microliters. So you really only want that red spot on the P1000 to be either 0 or 1. If you somehow manage to crank it above 1, this thing's going to get screwed up. So you want to be super careful with the pipettes. All right. Those are the pipettes, but so what the heck's a microliter? So when we're dealing with numbers, right, we can deal with really big numbers, we deal with really small numbers, but it's kind of hard to talk about, right? So if we look at this number here, this 1 with 14 zeros after it's 1 times 10 to the 14th, right? When we talk about this many zeros, it's kind of hard to deal with. So that's why we use these things called exponents, and that kind of helps deal with some of those numbers. So when we're dealing with smaller things like 0.001 here, we can call that 1 times 10 to the negative 3. We'll get back to that in a second. When we're always dealing with measuring stuff in the lab, there are certain units that we're always going to work with. Okay, so if we're dealing with volume, it's always going to be a volume measurement in liters. And we symbolize that as a lowercase or a capital L. They're sort of interchangeable. If we're dealing with length, not that we deal with length too much in this lab, we're dealing with meters. When we're talking about the mass of something, this can also sort of be considered the amount of something. We're going to be dealing with grams. When we're talking about the number of things, this is also an amount, right? It's a different way of talking about amount. We refer to that as moles. Okay, so 6.02 times 10 to the 23 of anything equals 1 mole of that thing. So if I had 6.02 times 10 to the 23 measuring cups, I would have 1 mole of measuring cups. When we're talking about concentration, we're talking about the amount per volume. We'll talk more about this in a second. A lot of the times when we're dealing with concentration, molarity is the main unit of measure, and that's typically a capital M, but there are other types of concentration that we deal with. When we're talking about molarity as a concentration, so if we have 1 molar here, the big capital M, 1 mole per 1 liter is 1 molar solution. Okay, so that's the concentration that we're dealing with. So again, we can go big or small when we're talking about these things. Most of the time when we're in the molecular biology lab, we're going to be in the smaller realm of things. And so if we're looking at a milli, okay, so these are modifiers that we're going to put on any of these units to sort of change what we're dealing with, okay? So when we're dealing with milli, we're talking about 1/1000th of something, okay? So we can refer to that as 1 times 10 to the negative 3, or we can refer to that as 0.001 of something. Conversely, if we talk about something like milliliters, if we have 1,000 milliliters, we have 1 liter. Okay, you can see how these two things are the same. And if we do the opposite of that, 1 milliliter equals 0.001 liters. Okay, 1/1000th of a liter equals 1 milliliter. And again, you see I have some as capital L and some as lowercase l. For liters, that's the only one that really you can switch with. Capital M and smaller m can have different meanings. So milliliters, milligrams, millimolar, those are things we can do, right? So I'm showing you liters here, but you can put that milli modifier on any of these units to talk about the small concentration of things. Micro is one millionth. Okay, so 1 times 10 to the negative 6, or 0.0000001 of something. That's a micro. So basically, if we had 1 million microliters, we would have 1 liter, or 1 microliter equals that many of a liter, 0.0000001, 1 times 10 to the negative 6. It's really small. That's why we need things like the pipettes to measure volumes like that. We typically don't measure nano in the lab, but it is possible. So I wanted to show it to you. So nano is 1 times 10 to the negative 9, or 1/1 billionth of something. So if we could measure 1 billion nanoliters, that would equal 1 liter. And if we had 1 billion nanograms, that would equal 1 gram. Conversely, 1 nanoliter is 1 times 10 to the negative 9th liters. Okay, so that many decimal places. Again, this isn't something that's very practical, but it does happen every once in a while. And we deal with things like nanograms sometimes, so it's useful to know. We can also go even smaller. So 1 times 10 to the negative 12 is a picoliter, or a pico. 1 times 10 to the negative 15th is a femto. So we can go even smaller than what we have here. The other thing to notice, right, we're going in sets of 3. Okay, so when we go from milli to micro to nano, we're going from 10 to the negative 3, 10 to the negative 6, 10 to the negative 9. So we're always moving 3 decimal places. And so that's why, when you're working in the lab, it's to me, it's kind of useful to think about this P1000. Right? A P1000 pipette can measure 1,000 microliters, or it can measure 1 milliliter. Okay, 1,000 microliters, 1 milliliter, it's the same thing. And so the microcentrifuge tubes that we have a maximum volume of 1.5 milliliters. And so it can you can sort of visualize in your head about what a milliliter is. And then if you were to divide that into 1,000, it's a microliter. So we can measure 1 microliter pretty reliably with a B20. And so you'll get the feel for what 1 microliter, what 1,000 microliters looks like. But it's useful to sort of have that visualized in your head. So when you're doing lab math and stuff, you can kind of say, okay, yeah, this makes sense. And like if you're measuring something out and it's like a big volume that you're only going to fit into a small tube, there's probably some sort of a math problem that's going on there. So when we have things like grams and moles, when we're talking about the amounts of things, the reason that these are sort of interchangeable in science is because each thing that we're going to measure kind of has a specific mass, and they're related to one another. So basically, if we have 1 mole of any pure atom, so if we have 6.02 times 10 to the 23 carbons, you'll notice that on any kind of periodic table, there's going to be an atomic weight to that molecule, to that atom. And so basically, if you have a whole mole of those things, that's the mass that it's going to be. So if we had 6.02 times 10 to the 23 atoms of carbon, it would equal 12 grams. Okay. So let's talk about an example here. So we have a little compound, sodium chloride, NaCl. If we go back here, you can see where's our Na. There's our Na. Our Cl should be over here. Yeah, there's some Cl. If we look at the atomic weights on that, 22.99 grams per mole. So if we have 22.99 grams of sodium, we have 1 mole of sodium. Chlorine, if we have 35.45 grams of chloride, then we have 1 mole of chloride. So how many atoms of sodium are in 22.99 grams? Well, to 1 mole. So that means we have 6.02 times 10 to the 23 atoms, right? So that's why we can say that mass in grams and moles are kind of equivalent, because if we know one, we know the other. But what about this whole compound? So sodium chloride, if we weigh that out, we have table salt here. So what would the atomic weight of sodium chloride be? And so I'm going to give you a second to sort of answer that in your head. How would we figure that out? We know we have this one. We have the sodium that equals that. We have the chlorine that equals that. Okay, so if we want to know the atomic weight of sodium chloride, we just add these two things together. So that means that, if we have 58.44 grams of sodium chloride, then that means we have 1 mole of sodium chloride. So now this relates to the concentration question, right? If we take that 58.44 grams of sodium chloride and put that into 1 liter of water, what would our concentration be? If we go back to this thing, again, molarity, concentration, the number of moles per liter, okay? Again, 58.44 grams equals 1 mole of sodium chloride. So we have 1 mole of sodium chloride going into 1 liter of water. So that would be a 1 molar solution of sodium chloride. Okay, so let's do that. Just want to point something out. So here I have 58.44 grams of sodium chloride. This is a graduated cylinder with 1 liter of water in it. I know you can't see it perfectly. If you look at this, the meniscus is at the measurement point. So the etched line is the measurement points for these things. If you have liquid in a graduated cylinder, it's generally going to be curved up at the edges that you want to be measuring from the lowest point of the curve. That's called the meniscus. And that's where you want to measure. So I have a 1 liter here. I'm going to add my 58.44 grams of sodium chloride into the here. And now I have theoretically a 1 molar solution. Okay. But if you've had chemistry, you should know that this is now actually not a 1 molar solution. I'm sorry, I know that's very confusing. So if you look at what happened here, when we added that mass into this 1 liter, it took up some volume. Okay, so if we look at that measurement point, our liquid volume is now above that measurement point. And so actually the right way to do a solution is you measure out a volume smaller than what you want, you pour in your dry stuff, you get that dissolved, and then you bring your volume up. Okay, so this is technically not a 1 molar solution right now. If we had put all that sodium chloride in, got it dissolved, and then brought the volume up to 1 liter, then that is a 1 molar solution. So this concentration is slightly off of 1 molar. Also, since we're looking at this is a 1 liter graduated cylinder. You can see it says 1,000 milliliters. So just to remind you that there's 1,000 milliliters in 1 liter. If we took our P1000 and set it to 1,000 and did this 1,000 times, all right, if we were putting 1,000, there's no tip on here, so you wouldn't actually be measuring this. If we measured 1 milliliter 1,000 times, we'd fill this thing up. Okay, so that's our working with concentration thing. Let's see. Let's go back to this. All right, so that is 1 molar, but what about if we want something smaller, like a 1 millimolar? Okay, and this is one of the things that people get confused about. So if we want a 1 millimolar solution, how would we go about doing that? Does that mean we change both the number of moles and the liters, or what? So we know 1 millimole right, would be one thousandth of a mole. So for sodium chloride, we just take that 58.44 grams, we divide it by 1,000. And so 0.05844 grams would be 1 millimole of sodium chloride. But do we dissolve that 0.5844 grams in 1 liter or 1 milliliter? And so think about that for a second, right? We want 1 millimolar. And so again, we can kind of go back to a liter. And if we want, this is 1 molar, how would we get 1 millimolar? So do we divide both sides by 1,000, or do we just divide one side by 1,000? Okay. So again, hopefully you've thought about that for a second. We've divided the top by 1,000 to get our 0.05844 grams to get 1 millimole of sodium. And we do put that into 1 liter. Okay, so whenever we're talking about millimolar, we're always going to just divide the top number, not the bottom number. So it's going to be, it would theoretically be that 0.05844 grams, a much smaller amount than what we had measured there to get into 1 liter to get a 1 millimolar solution. And the thing about concentration is we always want to think about it as a relationship. Okay, so sometimes, I've been talking about molarity. Sometimes we talk about concentration as, grams per liter. So when we think about something like this, I have 15 milligrams per mil here, 15 mgs per mil. We sometimes call these mgs instead of milligrams, just to make it easier to say. 15 milligrams per milliliter is technically the same thing as this. You can think of it as a fraction. 15 milligrams per 1 milliliter. Okay, this is the same thing as this. And with any kind of fraction, if we go back to algebra or basic math, if you divide or multiply the top and the bottom by the same thing, then they equal the same thing. And so if we change our units the same on both sides, then we get the same thing. Okay, so here's our 15 milligrams per milliliter. That's technically the same thing as 15 grams per liter. It's the same thing as 15 micrograms per microliter, 15 nanograms per nanoliter, right? Again, we're dividing both the top and the bottom by the same thing whenever we change both units like this. So grams divided by 1,000 is milligrams, liter divided by 1,000 is milliliters, grams divided by 1,000,000 is micro, liter divided by 1,000 or 1,000,000 is micro, grams divided by 1,000,000,000 is nano, liters divided by 1,000,000,000 is nano. Conversely, we can do some other equivalent things here. So if we have 15 mgs per, right? So we said we had 15 mgs per milliliter, right? We can also express milliliters as microliters, okay? So 1 milliliter is 1,000 microliters, right? That's equivalent. So 15 milligrams per 1,000 microliters is the same thing as this. Conversely, if we go the other way, if we go 15,000 micrograms, it's the same thing as 15 grams. Okay, double check that math in your head. Or sorry, yeah, see, I just double checked the math in my head and I did it wrong. 15,000 micrograms is the same thing as 15 milligrams. Okay, 15,000 micrograms. So if we divide this by 1,000, we get milligrams. 15,000 micrograms is the same thing as 15 milligrams, and that's per mil here. Okay. So we can change these units. We can make these sort of equivalent things just to maintain our ratios. This all sounds really confusing. And Obviously it is, but you're going to do a lot of math like that, like this year. So we're going to get a lot of practice doing it. If it's hard understanding on a screen like this, you're not alone. I didn't understand all this stuff until I actually started doing it. So, um, you know, be patient. We're going to work through this. Um, so I've been talking about molarity and then things like milligrams per milliliters, concentrations. Unfortunately, in the lab, we do a lot of other kinds of concentration measurements. One of the types of measurements we do is called percent. Okay, and so percent is either weight to volume or volume to volume. And we use this a lot when we're making agarose gels when we do DNA. And percent is just the number of grams per 100 milliliters. And so if we had 100 milliliters, if we had 100 milliliters of something in a graduated cylinder, not this one, this is 1,000. If we had 3 grams of something, if we had 3 grams of agarose that we were going to put into our gel, and it was a 100 milliliters of buffer, that would be a 3% solution. Okay, if we had 3 milliliters of buffer or detergent that we're putting in 100 milliliters of buffer, then that would be a 3% solution. So this is another unit that you have to deal with. And then there's this one that's actually really easy, but people overthink it. And so this is another one that you have to practice to get used to it. So this unit is called X. Okay, so it's how many more times concentrated something is than what we want to use it at. And so typically, whenever we see a solution like this, we want to use the final solution at 1x concentration. So our gel buffer comes as a 50x solution. We want to use it as a 1x solution. So that means it's 50 times more concentrated than what we want to use it at. When we're going to be doing some things called restriction digest. So that the buffer that we use to make our enzyme happy comes at a 10x concentration. We want to use that as a 1x final concentration. And so basically it's just telling you how much you need to dilute these things out. So example here, if you have a 6x buffer, you ultimately want to have 1 part of buffer and 5 parts of other stuff. Or if we have a 10x buffer, you want to have 1 part buffer and 9 parts of other stuff. And so if we have a final volume that we're trying to get to, right? So if we wanted to make 1 liter of something that had a 10x concentration, right? We wanted to get it down to 1x. We take that total final volume and divide it by whatever the x number is. Okay, so let's think about this liter as milliliters. Okay, so we have 1,000 milliliters that we want to make a total solution of. We take that 1,000 milliliters, we divide it by our x number. So if we say we're doing a 10x buffer, 1,000 divided by 10 is 100. And so we'd put 100 milliliters of our buffer in there. And then we'd put 900 milliliters of water to dilute it out. Okay, so that's sort of this one, right? 100 milliliters, 900 milliliters. The nice thing about X is you don't always have to have a final target volume. Sometimes you can be working with something, right? That if you already have a volume that you're working with and you're going to be putting a buffer onto it, you can figure out the math there. And so if you already have a volume and you want to put a buffer on there, you have to divide that volume by x minus 1. Okay, so in this example, we already have 1,000 milliliters of something. If we wanted to add a 10x buffer to that, we'd have to divide that 1,000 by 9. And then that would be the volume of buffer that we would add onto this to make a 1x solution. Okay. It's the two different ways. So one, if you're going to make a target volume, so if we're going to put 100 milliliters in here and then add water, that's just the total final volume divided by 10. If you're already starting, like what I have here, you'd take this total volume divided by 9, and then that's the volume that you're working with, or 9 being x minus 1 in this example. Okay, so again, this is something you're going to be working with this semester. You're going to get a lot of practice. Don't worry about if this doesn't make total sense right now. And so whenever we're pipetting, we're always going to be pipetting volume, right? But most of the time, we're going to be able to figure out, or we're going to know the concentration of whatever we're pipetting. So if we know the volume of what we're pipetting and we know the concentration of what we're pipetting, then we know the amount of whatever's in that solution that we're going to be putting in there, whether it's moles or grams. Okay, so there's going to be times where I'm going to ask you to pipette a certain number of micrograms. Well, you can't pipette micrograms directly, but you can pipette micrograms as a concentration. Okay, so we're going to know the concentration of something. You're going to have to do a little bit of math, and you're going to be pipetting the appropriate volume to get the amount of grams or moles or whatever into the pipette, into the microcentrifuge tube. Oh, one thing I forgot to mention. Let's put a tip on our pipette here. Whenever, let's stop the share. So whenever you're pipetting something and you have liquid in here, never turn your pipette like this. Okay, so what can happen is the liquid can come down and go inside. You know, you're going to get a little bit of the hydrostatic pressure keeping the things together. But, you know, you're going to want to pipette and always keep the pipette vertical like this. And we have racks that help, you know, make sure our pipettes are kind of sitting like this. If you bring something up into your tip and then, you know, like wander around, that liquid can go down in there and that kind of messes up the pipette. So, you know, always do your measurement, put it into your tube, and then, you know, eject your tip into the waste containers. Never flip them over. All right, so what are we actually gonna do this week? And so I know last week, or my last intro, I said we were gonna do a semester-long project where we're gonna clone a gene as a fluorescent fusion protein and stick that into cells. And you know, that's fun and cool. But the first week, so when I was a postdoc, and we would have students come and rotate in our lab, we would always test their pipetting skills, and they were always terrible at it. And we would test their pipetting skills by a technique called a protein concentration assay. And basically what this does is it makes you do a lot of pipetting. It makes you do a lot of concentration to make sure concentration as in focus, but you use this assay to determine protein concentration. And this is a really good assay because it kind of tattles on you and it shows when you make mistakes pretty clearly. And don't worry about making mistakes. It's fine. It's just a way to learn. So the main thing that we do here, and this little picture that you see is an example of the assay when we're done. This is a 96 well plate. Basically, it's like a lot of microcentrifuge tubes all crammed together into one plate. And so we're going to do something called a BCA assay. And it's a type of protein concentration assay. Essentially, what we do is we use a curve of known protein concentration to determine the protein concentration of a couple unknowns. Okay, so you can see here how this color is a gradient, right, as we go along here, and these are triplicates. As we get higher in protein concentration, we get higher in this purple color. Okay, so the purple color is equivalent to protein concentration. And then we can quantify the amount of color change that we have using something called a microplate reader. So this thing's a microplate. We have a reader that can measure how much light gets absorbed by each one of these wells. And that quantifies the indirectly, it quantifies the protein concentration. Directly, it quantifies how much light moves through each one of the wells. We have to do a little bit of math to go back. And so we do something called a standard curve of known concentrations. And then we have some unknowns. And what we do is we compare how much light gets absorbed in these unknowns to the known protein concentrations. We can say here, okay, this one is about this one. And so their concentrations are probably the same. Ultimately, you're going to graph this all out and it's going to, give you much more precise than, yeah, that one's about like that one. So we're going to have actual quantified protein concentrations that we're going to be working with. If you're into chemistry, I'll talk more about this later in the semester if we use this, but the BCA assay actually stands for bicinchonic acid. Essentially what happens is copper gets reduced in something called the Biuret's reaction. And when that happens, we get that purple color. And so the more protein that's in a sample, the more Biuret reaction we get, the more purple color we get. And so that's how this technically works. One of the confusing bits about this assay, right? I called it, it's the BCA assay for this word right here. We use a known protein concentration out of something called bovine serum albumin. Basically, it's a blood protein that can be purified pretty quickly and cheaply. And we buy an analytical standard that's at a known concentration. So we know that our bovine serum albumin is at 2 milligrams per milliliter, which is the same thing as 2 grams per liter or 2 micrograms per microliter, as I said before. But anyway, we buy it at 2 milligrams per milliliter. And then what we do is we dilute that out. And so as you do this, you're going to be pipetting different amounts of different volumes of the standard into each well. And that's going to give you your standard curve. And then we're going to compare the unknowns to the intensity of light that gets absorbed, or the absorbance of light. So to actually do this, going to schedule up a time for next week. I'd like you to sign up for a 2-hour slot on the sign up sheet. And that'll be to actually do the assay. And then once you have the absorbance, the intensity recorded, you can do the results working with the data at home. Or you can come back in and I can help you with that at another time. So generally, you're going to be pipetting so much that you're going to be kind of burnt out here, and you're not going to want to do all the math right away. So it's cool if you want to, do the assay and then come back another time, and I can help you with the results. At the end of this assay, in the general class forum on Moodle, I want you to post your Excel file and the concentration that you determined to be your unknown protein concentration onto that forum. Okay. And so that's what we're going to do for the first week. And again, that doesn't fit into our general flow, but this is a really good assay to sort of get you to learn to focus on what you're pipetting and keep records of what you're pipetting along the way. And it's going to be a lot of fun. So sign up using the sign up sheet posted on Moodle. And I'll see you when you come in.